From Microbe TV, this is Office Hours for Wednesday, May 1st, 2024. It's like Labor Day in many countries in the world. I'm Vincent Racaniello, and welcome to your viral corner of the internet. Joining me tonight here in the incubator is Karen. Welcome.
Hello.
People were wondering where you were last week. They were asking for you.
I totally forgot about you last week.
Actually, I don't have any volume from you, so give me a talk.
Check, check. Testing. Oh, now I'm very loud in the headphones.
Maybe it is. Maybe it was fun. Do you guys hear Karen? Talk a little bit, Karen.
Okay. Say that some lady, as perhaps there is, have for your love as great a pang of heart as you have for Olivia. You cannot love her. You tell her so. Must she not then be answered?
Oh, Karen.
Keep going.
Karen's
I got
good.
So much more memorized, if you want.
It's good. Thank you very much. All right. So, as you all know, I've been practicing some Russian. I have Privyet. And then at the end of the show, I'll say Poka. But tonight, our intrepid moderator, Vanity Nutrition, taught me Dobry večer.
No, that's not how you were saying
Dobry večer. I got the R's right, at least. Dobry večer.
Initially you said Dobry večer.
Dobry večer.
Yeah, that's what you said the first time.
Anyway, Dobry večer, everyone. Got the rolling tongue going there. Thank you, Vanity. Let's see who is here for moderating tonight. We have Andrew from New Zealand. We have Vanity Nutrition from Long Island. We have, I saw Steph from San Francisco, Tom Steinberg from Western Oregon. Anyone else?
Oh, Dennis says I'm a little weak.
Weak?
Yeah. I'm hoping he means the microphone.
I can turn you up a bit. There's about 4 notches. Try that. Where was I? Les from California. Welcome. And I think that does it for our intrepid moderators. Thank you all for joining us tonight. The moderators, thank you for joining and moderating once again. And many of you were here for the 1 p.m. TWEVO. Thanks for coming back. And all of you who are here this evening to partake in virology knowledge, welcome. We have a little mini, no, I shouldn't say little mini. It's a mini lecture. We don't need to have so many adjectives which say the same thing, but we tend to do that, right? So what's happening in the world of viruses? Well, my virology course ended technically on Monday, and now we have 25 lectures in virology for 2024, all on YouTube for everyone to partake in for as long as you want. All the years worth of lectures are up there. So if you have a lot of time, you could go through and see how they've changed over the years. You know, we used to have an H5N1 lecture. It was a big brouhaha in 2015. We had a lecture. Now maybe it's time to have another one. So next year that would be, we didn't have one this year. So that's cool. And it's next week is finals week. And then we have more time here at the incubator to make content. So before we go to mini lecture, tell me where you are from. We would like to highlight where you are coming from. I told you where our moderators were from. Vanity and I were exchanging voice text files so I could practice my Dobry večer. Dobry večer. Is it Dobry or Dobre? Dobry večer. Dobry večer. Oh my gosh. I lost your voice message, Vanity.
You said Dobry the first 1,000 times.
Dobry večer.
Yes.
So, good evening, everyone. Buonasera. Bonsoir. What's the Spanish? Do you know? You must know, Karen, Spanish.
Hola?
No, that's hello, right?
I don't know what you're saying in the other languages.
Let's say, I'm saying good evening. Good evening. Buenas noches. So those are I don't know any Spanish really, but the other languages I can speak, well, Italian and French I can speak Russian I can't speak at all. Those full words. So good evening and welcome, everyone. Where you're from? Kang is from Chicago, 20C and partly cloudy. It's also 20C and partly cloudy here in New York City. Lise, hello, Lise from Columbus, Ohio, 78 and sunny. Wow. It's your birthday? Happy birthday, Lise. If it's your birthday, you say it's that birthday weather. Cool.
You know, now we're looking at several people are using Russian letters. Yes? And I just realized something.
What's that?
Well, I had a terrible time. Somebody's name on their PayPal donation was written in Russian. And I'm going to probably next time I'll just bring their name and somebody can translate it for me because QuickBooks does not accept Russian letters.
It's funny. All right, Lisa's birthday. Happy birthday, Lise. Tom is from Western Oregon, as we heard. Maureen is in northeastern Ohio, another Ohioan. We have a lot of Ohioans. John is from Minneapolis. He is cooking a ribeye steak and zucchini squash onion, which he grilled over charcoal. Wow. Abdul Aziz is from Saudi Arabia. That's pretty far. Will is from Changsha, China. Patricia, where is Patricia from? Nice summer evening in D.C. Welcome. Abdul Rahman from New York City. Welcome. David is from Dayton, Ohio, our third Ohioan tonight. Wow, Ohio's likes viruses, don't they? Bernard is in Charlottesville, Virginia. Claire is in the UK where it's pretty late. 8, 9, 10, 11, 12, 1, 2 a.m. or something like that. I can never do the daylight stuff, so. Who else is here? Karen is a little weak, yes. Peter is from Boulder, Colorado, where it's 288 Kelvin and overcast. And Vanity, you like my R roll? Thank you. Well, you know, the Italians can do the R roll, so I guess it comes naturally.
There it's phonetically spelled out. Dobry večer.
Dobry večer.
That's what you said the first 1,000 times when you were practicing before.
Now, here's Abdur saying das vidanya. Yeah, that one I know, right? And goodbye, but we're not saying das vidanya yet. And Rima says something in Russian that I can't translate, so perhaps anyone who speaks Russian can translate that, or maybe Rima. Do you speak Russian? Lucy's from Sechelt, BC. Frank is from Santa Barbara. Merhaba. What does merhaba mean? Is that what you were translating? Rona is on Long Island. Betty is from Washington Heights, New York City, where I spent many years of my life. Marty is from Toronto. Older and Wiser is from Cleveland. Patricia is from Massachusetts. Lisa is from Sperryville, which is in Virginia. Mike is in Seattle. And John says, a Russian woman taught me this. Ya tebya lublю, la teba lubu. I don't know what it means.
Oh, I do. There's
What?
a song that I sang once, Ya tibia libu, or whatever, but that's pronounced wrong because I was in 7th grade and it means I love you.
Oh, that's cute. Well, a Russian woman would say that to you, John, wouldn't she? Jessica is from the University of Toledo, which is in Toledo, Ohio. Welcome back. Leo is in the North California coast. He's making chicken tacos. Rima's in Iowa, where the weather is crazy. Dobry večer. I said good evening enough now. I know you're going to get tired of it. Parallaxed is in the southeast UK, where the weather is dark.
As in, what, like dark?
I don't know.
I don't know. Now I have all
Well,
kinds
like
of
cloudy.
pictures.
Patricia says buenas noches, or just buenas. Buenas noches. Thank you for the help. Elizabeth is from West Virginia.
Where you say good evening by saying hiya.
Hiya. Is that right?
Hiya.
Oh,
That's how West Virginians say good evening.
hiya. And Riemann says guten Abend. What's guten Abend? Good evening.
I'm pretty sure everybody's just going to go with that.
Cheryl is from Santa Rosa, 79 and sunny. Oh, very good, Lisa. It is your birthday. Happy birthday. Happy birthday. And have a great one. Michaela is from Buenos Aires. Buenas noches from Buenos Aires. Cool. EHS is from Lehigh Valley, Pennsylvania. This is one of my most fun parts of the evening, telling you where you're from. Questella is from Ohio. I know that because Questella has been here since day one. Maybe some of you have also. MN is outside of D.C. Markle is from Eastern Long Island. Ohio is a TWIV stronghold. We should do the election there and TWIV will win. Oh, Steve is in Kauai. Cool. That's very cool. In the middle of the Pacific. Another Steve is in Columbus. There you go. Another Columbus, Ohioan. MN says, welcome back, Karen.
Hello.
Patricia writes, it's interesting to hear so many from Ohio. At the start of the pandemic, the state was fairly responsive and active. I visited in, I think it was March 2020, and things were just getting going, and they were just starting to have cases in Ohio. I remember that very clearly. When I went back last week, I stayed in the same hotel on campus there. Razor is from Saskatchewan, Canada. Yeah.
Oh,
Carol.
it looks like
What's that?
Lee says there's at least 6 people in the house from Ohio. And I'm wondering where in Ohio, because we're going to be in Ohio soon.
Well, some of them are in Columbus. One was in Toledo, and I don't know where the others are.
Well, we will have non-conference podcasts, hopefully, at Ohio
One
State.
hopes.
Mm-hmm. Which
So
people could come to.
Carol is, yes, stay tuned for that. Carol is from Southern California, our favorite nephrologist. Yes. Rima spelled out what you said. Yes, that was the thing. I said, what is that? And that's what she had said. Very good. If you use Google Translate, you can use the audio symbol to learn where to put your accent when speaking Russian. Okay, Rima says, it is dobro večer, dobro? No, she's phonetically saying dobro, dobry večer. Merhaba is hello in Levantine. Thank you. Elicio is from Brazil. Dennis says, merhaba is greeting in Turkish, I think. Logan is in Atlanta. Lalu, what a cool name, Lalu. I love Lalu. Great name. Is from Montana. Very cool. Joseph is in Southern Ontario. Janice is in Butler, Pennsylvania. Mr. Doreen is in Elgin, Illinois. He's using his phone. No internet. Well, we know that here at the incubator. We used our phone to do the live stream as a hotspot a couple weeks ago. It worked beautifully, beautifully. Stephanie, hola from Mexico. Walter is North Central Massachusetts. Pete's in London, where it's 1 a.m. Karen's pronunciation of Russian is not bad.
So far.
If you can say Nido Viridi, you can say whatever that is. I don't know what that is. James is in Longmont, Colorado.
I have family in Longmont.
Really?
Mm-hmm.
All right, that's it for now. That is the people who are here. We'll let the rest of you flow in, and I'll go back and take your questions. Abdul says maraba is hello in Arabic too. Thank you. So now we're getting the language. What do you call it? When something happens over and over again on a show, what do we call that? an arc. We have a language arc now.
I was going to
on
say
Office
obsession.
hours. I started saying things, and now we are here. Hakan says you can also use Wiktionary to find the accented word vowels in Russian. It will show the word with an accent symbol over the volume. Cool. Jill is from Minnesota. Freddie is Ronnie from Dearborn, Michigan. Welcome, everyone. Don't forget to hit the like button. You got 125 people here. What do you want to see me?
Yeah.
Why would you do that?
I moved the speaker so I can look at you.
She moved the speaker so she can see me from across the room.
See, I'm not weak.
No one said you were weak.
Yes, they did.
Nobody said. I never said you were weak.
No. Somebody said I was weak.
Oh, the voice. He meant the volume or the
I
gain.
know, I'm just kidding.
He said that was weak. By the way, I bought a bunch of cheap Amazon glasses this week. These are not cheap. These are professional. But I bought a bunch of cheap ones just to see what they would look like. And I really like these. Look, you see the thing on the side?
Well, let me see.
You can see it there.
Oh, very nice.
But they're a little small. I'll wear these to read, but just thought you would like to see them. That's another arc we have here on Office Hours.
Should we talk about audio equipment again?
No, these things only happen right once. Here we go. Krishnan is from Malaysia. Welcome. That is amazing. That's far away. Jeremy is in Atlanta. Patrick is in Bremerton, Washington. And I'm glad you didn't say Washington State, Patrick. You get a thumbs up for just saying Washington, because that's what it is. If you're in D.C., you say Washington, D.C. And so many people add the state on, and kind of bugs me, but not that much. I just complain about it because I need things to complain about, I suppose.
Do you know, Vincent, I'm from Indianapolis, and you would be surprised how many people ask me what state it's in.
What state Indianapolis is in.
Mm-hmm.
I don't have your audio in my headphones. I wonder why. But I'm not going to worry about it. Let's try this. Hold on. Talk.
Hello.
No.
Hello.
We'll work it out another time, because I can hear you across the room.
Ah.
It's no problem. Heather is from Kamloops, BC. Yeah, I know. There's this detective fiction writer. I can't remember her name, but she writes in different stories in different national parks, and one of them was Kamloops. And we have Kip and Laura from San Francisco. Thank you for your contribution to science communication. Hit the like button. We have 130 people and 72 likes. So please hit the like button, which will attract people. It's cool. I just saw Veronica is here. Let's get Very up. Here we go. Everyone was very surprised to join the stream at the here. Dobry večer. It's 3 a.m. here in Moscow. A nice spring night. Everything is blooming. Nightingales are singing. Yeah, we thought you would be here so you could hear that. I also said privyet. So it's cool. Tona says, I like Karen joining. Gives me Ed McMahon vibes. Indeed.
I'll take it.
It's funny that you're sitting over there. It's kind of like Johnny Carson, Ed McMahon.
Aren't you glad I moved the speaker? Now you can see me?
You had a nice glow on your face from the monitor. Yeah. Okay. Let's see this. One more from Richard. Before Washington became a state, they considered naming it Columbia after the river. People objected because it would get confused with the District of Columbia. Interesting.
More so than it already gets confused?
Colombia, but that's like the country in South America, Colombia, right? Anyway, folks, what I would like to do before I get to your questions is do a little mini lecture. So this is from my virology course, which just ended. So let's see how we can do the screen share here.
Can you make me a thingamajigger so I can reply to people?
A moderator?
Yeah.
But weren't you last time?
Yeah. Oh, wait, maybe it's because I'm in on my microbe instead of my personal. That's what's going on. Never mind.
If you log in on...
Never mind, just ignore me.
All right, so that's not quite big enough. Let's see. There's always this little delay. Uh, let's see how that works. The full screen. I want to see how the full screen looks. Oh, I forgot. I have to turn on the Vinny. Where's the Vinny? Here it is. We need to turn on the Vinny. Where's my picture, though? Why does it have a... Oh, I see. It's got a picture of the last chat in there. That's pretty funny. So now it's working, but where am I? Oh, I have to bring it up above the screen share. That's right. I learned that last time. The things I learned, Karen, and I just keep learning and learning. All right,
Over and over
today,
again.
this is part one of a lecture I gave not too long ago in the course, which I called The Tale of Two Pandemics. And it's about HIV, AIDS, and COVID, SARS-CoV-2, COVID. But I'm just going to talk a little bit about HIV, AIDS origins today. I want to talk just about where it came from, because it's another virus, like all viruses, that came from nature. And when HIV emerged in the 80s, people blamed it on being made in a government lab in the U.S. No kidding. They did. And until we found where it came from, which I'm going to tell you about today, that's what people thought. So this is a tale of two pandemics. And I do like the quote here, nature is not human-hearted. Okay, where's my stinking cursor? Look, it's on the right-hand screen. I want it on my screen here. Let me go to the preferences. I need a cursor. There's this thing that says...
It's wiggling around on there.
Show pointer when using the mouse or trackpad. Show pointer only on slides with links. No, I don't want it only on slides. What stupid selections, defaults? There it is. There's my pointer. Nature is not human-hearted. Lao Tzu. That's true. Nature is not human-hearted. Neither are humans. So first of all, what is a pandemic? Oh, I have two definitions here. The first is kind of the sciencey one. It's an epidemic occurring worldwide or over a very wide area, crossing international boundaries, and usually affecting a large number of people. That's a pandemic. So what does affect mean? Right? Affecting is kind of vague. So here's a definition: an epidemic occurring worldwide that affects how we live, work, or play. I kind of like that on its own, but it may not apply all the time. But between the two, we capture it. And just to remind you, seasonal epidemics are not pandemics. So every winter we have a flu season. It's not a pandemic. Not a pandemic. And this definition tells us nothing about population immunity, virology, or disease severity. The main reason for saying we're in a pandemic is the WHO wants to get countries moving and do things to save people's lives. That's why they say we're in a pandemic. So the interesting thing about the HIV/AIDS pandemic, but which, by the way, we are still experiencing, we are still in it. It is many millions of people infected. You'll see the numbers in a moment. And we can slow it, but we haven't stopped it. This book is really good. Slides are cut off a bit on the left, yeah. I wonder why. I should say if I fit, it's what I have here. I don't know why. I see it's cut off, yeah. So maybe I'll just stick with the not full screen thingy. I hope that's going to be big enough for people.
You can stretch it some more without.
Let's try
blowing
to
it
go
all the
to
way up.
75%. Yeah, it can go a little more to 100%. Nope, 100% breaks it. 75%. I hope you can see that, folks. Yeah. Is this good for you? Let's get rid of the notes at the bottom. Let's see how that show. Where's the notes? Hide. Forget it. I can't get rid of this thing at the bottom here. Oh, there we go. So you should read this book, The Origin of AIDS by Jacques Pepin. He was a physician who worked in Africa for many years and had a good view of what was going on. And he writes in the book, this tragedy was facilitated or even caused by human interventions, colonization, urbanization, and probably well-intentioned public health campaigns. You'll see what happened here. So this begins in 1981 in June with a report in Morbidity and Mortality Weekly Report on pneumocystis pneumonia in Los Angeles. 5 young men, all active homosexuals, or today we would say men who have sex with men, were treated with biopsy-confirmed pneumocystis at 3 different hospitals. 2 of them died. All 5 had confirmed previous or current cytomegalovirus infection and candidal mucosal infection. So this is very unusual. Pneumocystis pneumonia in the U.S. almost exclusively limited to severely immunosuppressed patients. These were healthy young men. They shouldn't have been getting this. And they say all the above observations suggest the possibility of a cellular immune dysfunction related to a common exposure that predisposes individuals to opportunistic infections. And they didn't know what it was, but it was clearly an immunosuppressive thing. Now, at the same time in New York, at Albert Einstein Medical College, a pediatric immunologist by the name of Ari Rubinstein, he's treating 5 black infants who have signs of severe immune deficiency, including pneumocystis pneumonia. At least three are children of women who use drugs and engage in sex work. He recognizes that the kids are showing the same illnesses affecting the gay men, but his colleagues said, Ari, you're wrong. You don't know what you're doing. These babies don't have anything related. But this was the first clue that this was not just a disease of men who have sex with men. These were babies who were getting it. And so this is just an example of the pervasive bias that ran throughout the beginning of this disease. This disease was called AIDS in 1982. It was formerly called GRID, gay-related immunodeficiency, a horrible name, because as I just showed you, it's not simply gay-related. It was called AIDS, acquired immunodeficiency syndrome. There were clusters of pneumocystis carinii and Kaposi sarcoma, this cancer shown here on the right.
Can I ask a question?
Yeah.
Was there a reason to believe at the time that sexuality had some bearing on it, like as opposed to sexual activity?
Most of the initial cases were in men who have sex with men. That was the problem. But they weren't the only ones, right?
Right.
But they were the predominant cases were there.
But was there a reason to believe that it was something that would only affect men?
Well, that would not really make sense, would it?
No,
because,
you would think scientists would know better and not call it.
Well, scientists never do, because when you look at the Mpox outbreak, it was the same thing, right? People said, oh, this is a gay disease, but it wasn't. It happens in heterosexuals as well. So you would think scientists would know better, but they don't.
You'd have to acknowledge first that there's a biological difference somehow in people that have sexualities that are different, right?
Right.
That's weird.
So the CDC established a case definition for AIDS, Kaposi sarcoma or opportunistic infections, very simple. And then subsequently found to be transmitted at birth and heterosexually and also by blood products. So many other modes of transmission. The virus was isolated in 1983 from the lymph node of a patient with swollen lymph nodes, lymphadenopathy in Paris. And that was done by Montagnier and Barré-Sinoussi, and they got the Nobel Prize for that in 2008. So that allowed a blood test to be developed the next year, which of course could be used to screen people and blood products, and then sequence of the genome and electron microscopy showed it to be a lentivirus, a known retrovirus. Now you have to remember, 1984, sequencing is still pretty rudimentary. I mean, I sequenced one of the first animal virus genomes in 1981. It took me a year to do that, so this is still quite early on. Just to show you where HIV sits among the retroviridae, here it is down here. It's in this subfamily called orthoretrovirinae. But we have Rous sarcoma virus in here. We have HTLV1, 2, and 3, walleye dermal sarcoma virus, which we talked about when we talked about transformation. And we have Maloney murine leukemia virus. So there's HIV 1 and 2. So we have 2 groups now of human retroviruses. We have the immunodeficiency viruses, HIV-1, and as you'll see, HIV-2 as well. And then the lymphotropic viruses, HTLV-1, 2, and 3. Human T-cell lymphotropic viruses, 1, 2, and 3, and there are 4 now. So lentiviruses, of which HIV and 1 and 2 are a member, they're not new. They're not unique to humans. There have been others studied, horse lentiviruses, for example, isolated in the 1900s. There are bovine, feline, and caprine immunodeficiency viruses, all related. So this wasn't a new lentivirus. It was a new one for humans, though, for sure. And so the name of the disease was called acquired immunodeficiency syndrome, or AIDS. And the virus was given a different name, HIV-1, human immunodeficiency virus 1. So, in fact, it had been named HTLV previously because at the time there were only 2 HTLVs, and Robert Gallo thought it was HTLV-3, but that was wrong. So it's HIV. And, this is a syndrome. It's the occurrence of a group of symptoms together. HIV causes AIDS. There's no question about it. There are many AIDS denialists out there who say, prove it to me that HIV causes AIDS. Well, it's been proven over and over by giving people HIV-contaminated blood. Very early in the outbreak, the blood centers were not checking blood, and they infected many people, unfortunately, and gave them AIDS. Let me give you a sense of the numbers before we talk about origins. This is really big pandemic. So here in the U.S., the virus has killed over 600,000 people, right? More than all the wars that we have fought in the 20th century. 1.1 million people are living with HIV infection, and 1 in 7 don't know it, and that's dangerous because they can spread it inadvertently. In 2019, the last statistics that I got here, 36,000 new infections, and this is a few hundred per minute. And in 2019, 69% in men who have sex with men, 23% in heterosexuals, and 7% in intravenous drug users. And the racial breakdown you can see in the U.S. is shown here at the bottom right. And also on the left is the age breakdown. So you have 13 to 24. The peak is in 25 to 34 years of age, and it goes down all the way up to 55 and older. In terms of global numbers, people living with HIV in 2022, 39 million globally. 630,000 people died in 2022 of HIV-related illnesses. And so this breaks down. The vast majority of infections are in adults, 37.5 million, women, 20 million, men, 15 years of up, 17 million, and then children, 1.5 million as well. So this is the breakdown by region. WHO divides the world into 8 regions. And so the vast majority of cases are in Eastern and Southern Africa, and far fewer in West and Central Africa. And then the rest of the world, you can see far fewer as well. And finally, who is getting it? Global numbers in 2020, 11% sex workers, 9% people who inject drugs, 23% men who have sex with men, 2% transgender people, and this 20% clients of sex workers and sex partners of other key populations. And then 35% is everyone in other different categories that don't fall into one category. So we can control AIDS. We have triple drug therapy now, 3 different antiretroviral drugs in a single tablet. And this has slowed the pandemic in many countries. But as you might guess, countries without money, it's harder for them to get this drug that they need. For example, in the parts of Africa where it's most frequent, it should be there in greater numbers, but it's not. But there's no cure. You have to take these antiretrovirals for the rest of your life. You cannot clear virus from an infected individual because the virus established itself in long-lived memory T cells, and you can't get rid of it. There's no vaccine, so we can't block that primary infection. You have to take antiviral drugs your whole life, and this, of course, leads to drug resistance, and the drugs also are expensive. So we have work to do for preventing AIDS. We're not anywhere near there yet.
Vincent, do you know anything about PrEP?
Yes, pre-exposure prophylaxis. So the drug that I showed you here is post-exposure prophylaxis. There's also pre-exposure where you can take a pill, a triple therapy, before you engage in risky activity, which will reduce your chances of being infected. So for people who are at high risk for acquiring infections, then that is available to you, which will reduce your chances of getting infected.
And what is it? an antiviral?
Yeah, it's 3 antivirals together,
Okay.
just like that one there. Yeah.
I was under the impression that if your viral load is low enough, that it's undetectable, which can happen with
Mm-hmm.
successful medication,
Right.
that you can't really spread it either.
Right. You have to well, you have to make sure you're taking the drugs all the time. You can't skip them because as soon as you skip, the virus comes back. And if you have developed resistant viruses and you
Wait,
don't
you just
know
said
it,
the virus comes back.
right?
So
Because
it goes away.
it doesn't ever go away. It resides in your memory T cells.
Right.
And on antiretrovirals, the viral load is very low. There are also people who naturally can control infection. They're called elite controllers, and they have almost undetectable viral loads in the blood. But you have to take these drugs. And yes, if you're taking the antiretrovirals and they're working, you're not likely to spread the infection. All right. Let's talk about where it came from for the rest of for the next 10 minutes or so.
I thought it was. Wait, wasn't it made by the government in Bethesda?
Yeah, the U.S. government.
In the
That's
Bethesda,
right.
Maryland. That's what I heard.
That's right. So when HIV/AIDS was discovered, people began in many countries to look at seropositivity of at-risk populations. And the first studies done in Africa, in Zaire and Rwanda, showed that AIDS was common in the 2 capitals, Kinshasa and Kigali. 90% of sex workers were infected. 90%. Oh my gosh, this is something that people didn't even know about forever until the 80s. And here it is. And that high number suggests that it may have emerged here because that's how you get such high seropositivity when it's just emerging everywhere else. Then they began testing archival serum samples that had been stored for many years. And this suggested that the virus was present in the 60s and 70s at multiple places in Central Africa, which we're looking at here, but not in Western or Eastern Africa. And a serum sample from a 1959 DRC adult male was positive for HIV-1, found in 1998. They could sequence the whole genome. Same thing from a DRC female in 1960. So 2 early HIV-positive samples, 59 and 60. And those 2 viruses differ by about 12%. 1 year, 12% difference. That's quite a bit. And it suggested that the virus had been circulating for a long time in that region. So the virus is clearly present in, at the time it was Leopoldville. Now it's Kinshasa, by 1959 to 1960. So where did it come from exactly? Well, the source came from studies done by Beatrice Hahn, shown here. Actually, this is a photo taken in my laboratory when the wall of polio used to be freestanding. And it was just between 2 lab benches. And that was never a good idea because people knocked it over all the time. But anyway, Beatrice Hahn, a virologist at UPenn, she decided to look in chimpanzees. Why did she do that? Well, in 1989, they isolated a related virus to HIV called simian immunodeficiency virus from a chimpanzee. It's called SIV-CPZ. And so Beatrice said, well, let's see if this was the precursor of HIV. So she collected chimp fecal samples from 90 sites throughout Central Africa. And those sites are shown here. Each of these circles is a colony of chimpanzees. They tend to be quite isolated. And there are multiple species of chimpanzees, right? Pan troglodytes, and then we have Verus elioti, troglodytes, schweinfurthi, and then we have the bonobos, Pan paniscus. Only PTT and PTS have SIV-CPZ, and those are these yellow circles on the western area and in the Central Africa there. So these chimpanzees have SIV, and it's among chimps. SIV is transmitted by sexual intercourse from mother to child, and possibly during aggression when they fight by blood spilling. Right, so very similar to human.
And if they use intravenous drugs.
And if the chimps use intravenous drugs, that's right.
Only if they share needles. It's not actually the drug.
They don't use an autoclave.
Mm-hmm.
Transmission per probability per coital act among chimps, and don't ask how they got this number, 0.008 to 0.0015 per coital act, and that's similar to in humans, 0.0011.
Okay, now wait. What do you mean don't ask how they got this fact?
Well, how would they know how many times chimps are doing it and whether they got SIV? How would
I
you
think
do
that's the most important question here.
I don't know how they did it actually, but it's an interesting statistic. Now, this number in humans, 0.0011, I told this to my class. Every year I tell them. And one year someone came up and said, "Oh, so I can tell my friends that they can just have sex because the frequency is so low." I said, "No, the first time could do it, right? It's just 0.0011 is a probability, and many people get it the first time." Anyway, in chimps, it's also pathogenic, and they get an immunosuppressive disease very similar to AIDS. So this is the ancestor of HIV-1. Let me show you how this worked. So here are people on the right here, and there are 4 groups of HIV viruses, M, N, P, and O. The M and the N groups are spillovers from a chimpanzee. At least one of them, at least the M is in Cameroon and the N elsewhere. And the P and the O are spillovers from a gorilla. Now, SIV is not a natural virus of gorillas and chimpanzees, nor of humans, of course, but that's SIV. Chimps and gorillas got the virus from Old World monkeys. So in Africa, there are many species of Old World monkeys. Maybe you've heard of some of them: mandrills, vervet monkeys, Mona monkeys. Each Old World monkey has its own kind of simian immunodeficiency virus. And they have little letters next to them because it tells you what species of monkey they came from, like SIV Mon for the Mona. And these SIVs are not pathogenic in the Old World monkeys. Apparently, they've been infecting them for thousands of years. The chimp got SIV from both a Mona and a red-capped mangabe. So somewhere a chimp was co-infected with viruses from both. They recombined in the chimp. We know this because we can do genome sequence analysis. And then that SIV CPZ was transmitted to humans on 2 separate occasions. And the same happened with chimpanzees, with gorillas. The gorilla actually acquired SIV from a chimp, the same recombinant, and probably in fighting. And that was transmitted on 2 separate occasions to people. So when did this happen? There were 4 separate spillovers, right? M, N, O, and P. M and O are the first 3 decades of the 20th century. So let's say 1920-ish. I like to put that number down. N and P are more recent than 1920, but we don't have enough data to pinpoint it. And, how did this happen? How did this virus infect people? Well, the main theory is that a hunter was looking for bushmeat, perhaps caught a chimp and was butchering it in the field and cut him or herself, and the virus from the chimp went into its blood.
Okay, I'm sorry. I have a serious question here. I
Yep.
used to, like 20 years ago, 30 years ago, at some point, I did about a decade of HIV awareness interactive theater in the schools and in the prisons.
Yep.
And it was our understanding at that time that cutting yourself because blood flows out, not in, isn't a real risk.
Oh, it is a risk. If you're butchering a chimp and you get a deep cut on your palm, say, the blood from the chimp will go into it.
But how long is it exposed to oxygen before the virus dies?
Well, it would take some hours before the infectivity is lost.
Really?
Yeah.
See, they told us then that.
No, this is a
exposed to-
definite risk, this bushmeat hunting.
Interesting.
So in that book, The Origin of AIDS, he does calculations, and people believe that in 1921, there were fewer than 10 people infected with SIV-CPZ from all this bushmeat hunting, because it wasn't just one person doing this. But only one spread and multiplied. And probably there have been many cross-species infections like this before and since, but this one spread like crazy. And why is it? Well, it has to do with the colonization of Africa. For example, Leopoldville was a fast-growing city in the region. Here's the population growth of these cities from 1880 to 1970, in huge numbers of population growth. And here in the beginning, when it's just getting going, that's the M group origin, like from 1890 through 1920, right? So it coincides with population growth. a lot of people went from the countryside into these big cities. And so one idea would be the cut hunter went to one of these cities, visited a brothel, caught a sexually transmitted disease, went to an STD clinic, and then it was amplified by the use of non-sterile syringes, by sex. Some women had 1,000 clients a year. That's a good way to spread it. Now, as you may know, much of Africa was taken over by European countries in the form of colonization at the beginning of the 19th century. Those countries, and here are all the European countries that took pieces of Africa for themselves. They established large population centers. They moved adult males from the country into these cities for labor. They had large-scale prostitution. They had health care. They had clinics to treat sexually transmitted diseases where they injected drugs without sterilizing the needles. And so we think this was a major factor in the amplification of HIV. And as an example of how you can spread an infectious disease with a non-sterile needle, at the beginning of the 20th century, Egypt wanted to eliminate schistosomiasis from its population. So it injected millions of people with an anti-parasitic drug, but they didn't sterilize the syringes. They gave millions of people hepatitis C virus. And Egypt is one of the highest seropositive populations in the world for hep C at the moment. So this is a plausible way for HIV-1 to have spread. Now, a number of years ago, Michael Warraby, who you may remember from COVID Origins, collected a large number of archival HIV samples and did a phylogenetic analysis, which gives us a good indication of the timeline of entry of the virus into the U.S. So it enters Haiti around 1969. So why Haiti? Well, there's a story behind that. The Congo was taken over by Belgium during this colonization of Africa. And in 1960, they said, we've had enough of you Belgians, get out. So they kicked out, the Congolese kicked out the Belgians. However, all the doctors were Belgian. So now the Congolese had a problem. So they reached out to Haiti for physicians because they spoke French. And that was the language in the Belgian Congo. And the physicians came and took care of patients, and they got AIDS, they got HIV infection and went back, and they brought it to Haiti, their home country, in 1969. It then goes from Haiti to New York around 1972. Haiti is a vacation destination, so many people are traveling there, acquiring HIV, comes to New York, and from there it goes to California and to other parts of the U.S. And you can see by this timeline, here's the Haitian virus that's kind of this green color at the root of all the others, right? It's the ancestor. And then you have the New York City viruses and San Francisco and so forth. And I just want to point out patient 0 here. You may remember a flight attendant by the name of Gaetan Dugas, who famously was infected very early on and traveled the world and had thousands of sex partners and spread the virus to many of them. And he was always thought to be patient 0, right? The first patient in the U.S., but he wasn't. You can see that some earlier patients have that honor. So from Africa to Haiti to the U.S., that is where HIV came from. And in spreading to many countries, of course, not just the U.S., the virus diversifies. So I told you there are 4 groups, right? 4 separate spillover events, 2 from chimps and 2 from gorillas into humans. But the group M is the most numerous. 99% of all HIV infections are group M. And as they multiply, they're diversified into subtypes, kind of like COVID, SARS-CoV-2 variants. And the group O stands for outlier, is less than 1% of infection. This is mainly Cameroon, Gabon, and neighboring countries. And then Group N is only 13 cases, also in Cameroon, and Group P, each 2 cases. So each of these 4 groups is an independent transmission of HIV to humans. So this has happened 4 times. And it probably still happens now, except that it's just harder for it to get a foothold in people. And finally, one last word. There is HIV-2 as well. This was first isolated in Guinea-Bissau. It has about 30 to 40% identity with HIV-1, restricted mainly to Western Africa. It is less virulent than HIV-1. Most infections don't progress to age, the immunosuppressive state where you die of opportunistic infections. It's less transmissible, and there's no mother-to-infant spread of this virus. This virus is a crossover from the sooty mangabey directly into people, right? So before, these old world monkeys shared their SIVs with chimps and then with humans. But here, the HIV-2 comes from either sooty mangabeys or Sykes monkeys. And this crossover has happened 8 times. So there are 8 distinct lineages of HIV-2, A through H, which each came from a distinct spillover into humans. You may ask, well, what's going on? Well, many people keep these animals as pets. They capture them, and they're wild animals. They have SIV, and then they transmit it to people, and that establishes the virus in the human population. So 2 examples of how a new human virus, HIV-1, HIV-2, 2 new human viruses originate from animals. There's no need to invoke laboratory origin.
Is there no mother-infant spread because it isn't transmitted through breast milk?
I think the virus titers are so low that it can't spread that way. Yeah, I think the virus titers in the blood are very low. And so I want to end with just this picture that I made years ago, which shows this chimpanzee, the origin of group M HIV. So SIV spilling into patient 0 somewhere around 1920, let's say, the cut hunter. It becomes HIV, a human virus. And so far, we've had 75 million infections and 32,000 deaths. And so that's what we're dealing with now. Okay.
I'm sorry to belabor this point, Vincent, but the internet is still saying that HIV dies within seconds.
No, it doesn't die within seconds. Sorry, it doesn't die within seconds.
Why is it saying this?
I don't know, but you have to. If you want to know the answer, you have to ask a virologist.
Okay.
Okay, let's go back to some questions. It certainly is labile, more so than other viruses. It's easily inactivated by drying, by low pH, by sunlight, and that's why in a way, it has to be transmitted by such close contact, intravenous inoculations or sex, because it won't spread outside. It's not spread by respiratory routes. It's not spread by contact, but it does last. It lasts more than seconds. Yeah. So let's see what questions you folks have. Pete says, in the early 80s, I was involved with a printing co-op, which had business with the Terrence Higgins Trust, the early AIDS support charity. Their early leaflets still called it HTLV3 before HIV. Yes, that was Robert Gallo's fault. Yeah, but it's no longer, of course. John says, update to my parasitism journey. Now, he's not having a bunch of parasitic infections. He's watching TWIP. He made it to the video versions and on TWIP 194. Looking forward to TWIM or immune as the next step. Not sure yet which it will be. So TWIP came after TWIV, and then came TWIM. So I would start with TWIM next. And after TWIM, it was TWIVO, and then immune, so that you know what order you should. You don't have to do that, obviously, but you could get a sense of what's going on in our minds. Okay. Karen is a little weak. Vincent, would you be able to give me the title of Dixon's Parasite book that was at the start of Parasite episodes? Parasitic Diseases. So he's written a bunch. The textbook is called Parasitic Diseases. All right. And you can find it online. They have a PDF version, which is free at parasiteswithoutborders.com. I think that's what you mean. He's written a couple of others, but that's the textbook on parasites. Seconds, huh, Karen? Wow.
Hmm?
Seconds.
Seconds what?
Seconds in activation. That's pretty fast of HIV. I'm just filling in the silence as I'm looking for another question.
Ah.
Frank writes, given that many findings that work with other animals do not work with humans, do you think the opposite might also be true? Things that would have worked with humans are not tried because they did not work on the other animals. I don't know. I don't think we go that direction very often, right? Usually we treat humans. We don't do experiments in them all that often. We make observations, of course, but I don't think we design animal strategies based on humans. We do animal trials if you want to make a vaccine or a drug for animals. You do trials in animals for that reason. Yes, you can't take a drug that works in people and just give it to animals. So, for example, what is it, remdesivir, which works well for SARS-CoV-2 infection in humans, apparently works for cats with certain coronavirus infections. And so people are, and that's by testing it in cats, right? And it turned out to work. And people are trying to get that licensed, but it doesn't seem to be going well. Everyone is saying hello here. We have to get through that. Steve, thank you for your contribution to science communication. Really appreciate it. You can go, that's a super chat, by the way. That's a little button below the chat, but you can also go to Venmo or microbe.tv/contribute. Mike, I like the red glasses, but I really like your expensive ones too. Good. I'm glad you like them. As David writes, has anyone done a comprehensive search for the furin cleavage site and viruses of animals that were sold at the Huanan wet market just before the pandemic? Well, there didn't have many samples from the wet market because on January 1st, 2020, the market was closed, the animals were removed, and the place was sanitized. So there were no animal samples, very few, and there were a few environmental samples, all of which are reported in the Michael Warraby papers. And they're only small pieces of sequence that allow you to say, yeah, this was SARS-CoV-2 early on. So we don't find the furin cleavage site in those at all. What are my thoughts on cyclospora? Well, what do you want to know? Anything in particular? We talk about it on TWIP a lot, right?
How do you feel about it?
How do I feel? I feel that it's an apicomplexan parasite. And you can get infected. That's what I feel, that it's an apicomplexan parasite. How would you get, you shed it in your stool and you can get infected by drinking water or eating food that's contaminated with it. And you have to watch out, especially in countries where the disease is present, that you're careful about what you eat and drink. But I feel that you should be careful. I don't think you should get infected. It's going to be an unpleasant situation. How do I feel? That's very funny. I like that. How do you feel about it?
They asked your thoughts. I'm more interested in how you feel.
Les says, every house in Washington, no state, has a plaque. George Washington did not sleep here. That's funny. I like it. Oh, Patricia writes, and by the way, folks, we do not give medical advice on office hours. The only medical advice you will get is on our podcast where Daniel Griffin is present or some other medical doctor. So Patricia writes, a friend got her first shingles shot. Doctor said 2nd in September. Should it be sooner? Her husband tried one shingles shot before COVID hit, didn't get the second. Would he have to get 2 shots again? 4 to 6 months apart is the recommended timing. And if you've delayed, you don't need to get 2 more shots again. You can just get that second one. So that's one of your favorite lectures, the HIV/AIDS. It's a really cool story of where the virus came from, right? It was all about that chimpanzee sampling in Africa by Beatrice Hahn. And, they collected feces, but also urine. It turns nobody asked, why is the virus in feces and urine? Well, it's just incidental, right? And they would pick up fecal samples from the forest floor, and they could tell which chimp it was because they did mitochondrial DNA sequencing, and that is individual specific. And they could say this is Betty and this is Joe and get multiple isolates from each individual chimp. They also collected urine in the morning when the chimps sleep up in the trees. And the first thing they do when they wake up is they pee and down to the forest. And they would stand there with collectors and collect the pee. Yes.
No.
Yeah, Beatrice told us this story.
No.
And then you could do mitochondrial typing. Would you like that job?
I feel like that would be like you have to run to like make it hit your cup.
Yeah. You have to, because remember, initially it's just coming over and you got to get in the right place and make sure you don't get it on you and so forth.
Wow, that sounds like an athletic job.
They had long poles with with collectors on the end. Yeah. I thought that was great. I really liked that.
I fantasize about the collecting poop jobs that you talk about all the time also.
Andrew says, my favorite plaque in London read, in 1779, Benedict Arnold lived here, patriot. It is all relative. Yes, indeed. Okay. Yes, I heard in the news that Professor Zhang Yongzhen in China has been locked out of his lab. He sent the genomic sequence for SARS-CoV-2 out of China in 2020. That's absolutely right. So there's an article in Nature, which we're going to highlight in TWIV on Friday. He sent Eddie Holmes the first sequence that he had derived of SARS-CoV-2, and Eddie Holmes said, "You have to do this. This is important for the world." And he said, "I'm going to get in trouble," but he did it, and he got in trouble. And he's been sleeping on the street apparently, but I'm not sure what's going on. They said they told him they were renovating his lab and moved him out, but he doesn't sleep in his lab. So what is he sleeping on the street for? I don't get it. There wasn't any detail in that article, so I don't know. But,
Vincent, Tony just said golden monkey showers for science.
Golden monkey showers for science. That's right. That's what it is. Yeah, you're going to get some pee on you for sure. You can't avoid it.
Do they, like, the scientists do that themselves, or do they hire, like, pee collectors for them?
I think some scientists like to do field collection themselves, for sure.
I was going to say, if they hired them, then, I might.
Yeah, well, there's a lot of sampling opportunities out there. Would you like to go sample bats in a cave?
Yeah.
You could do that. But I need you here at the incubator.
All right, never mind.
All right, Hat says, so Peter Daszak in front of Congress. Congressmen women were hilarious sad because they were so lost on the science. When they let him talk, he made them look even sillier, smaller. Yeah, well, I interviewed Peter Daszak in Singapore in December 2019. And that video where he explained how they were doing recombinant work in Wuhan has been used by many news outlets to say that this is how he made SARS-CoV-2, which is ridiculous, right? It's not how he made it. There's no relationship. But he's a smart guy, yeah. Too bad things have happened. Okay, Veronika, Privyet. I recently learned that scientists found another viral remnant in our DNA. Apparently, ancient retroviruses helped vertebrates develop our complicated nervous system. Perfect for twin. So the Jason Shepard, he discovered that a retroviral gag protein called ARC is a neurotransmitter in the nervous system. So maybe this is another example of how a endogenous retroviral genome is somehow participating in that. Yep, very interesting. Oh, dear. Have you heard of highly pathogenic avian influenza in dairy cows? I have heard certainly of avian H5N1 influenza virus in dairy cows. Now, I was told today, don't call it highly pathogenic because it's highly pathogenic in birds, but it's not highly pathogenic in cows. And this is true. The cows don't get very sick. But yes, it is infecting cows throughout the U.S. And in Ohio, where I was recently, many cows have been found to be infected. As you know, there's been at least a couple of human infections. But yes, and this is a bit worrisome because the more animals the virus infects, the more likelihood it is to change properties and maybe transmit among people. But as I've said before on this stream, we are pretty well prepared for this if it breaks out compared with SARS-CoV-2. We have 3 antivirals, we have vaccines, and we can make vaccines relatively quickly. So I'm not losing sleep over it. Let's put it that way. And Elizabeth says, this is absolutely true. This comment came up when we were talking about how scientists initially called AIDS GRID. And she writes, scientists are people like everyone else. Karen will disagree with you, but let me finish reading. We have all the same prejudices and blind spots to varying degrees. We have to remind ourselves of that. Now, Karen, you don't think scientists are people, right?
No, I do. I just think you sometimes need to act more like a human in front of people so that they remember that you're a human. That's all.
How do I not act like a human in front of? I'm just curious.
By doing like human people things.
No, what do I not do that I should be doing?
Oh, I don't know. A lot of what you do is science talk. That's all I'm saying. So I think it's easy for those of us that are not scientists to forget that you're also a human. Does that make sense?
Yeah, I do. But
it's not an insult.
There's no question that I use science as my launching pad, right?
Right.
Because that's what I know. But now you can see I can shoot the breeze with people on different topics, right?
Yeah, you can list off places in the country.
Yeah, right. Indiana. What state is Indiana in?
It is in Indiana.
But yeah, scientists are people. They have the same prejudices, and often they don't even know it. Yeah. Les says, Gallo first claimed to have isolated the virus. That's correct. But it turned out he had the Pasteur Institute isolate. That's right. He just re-isolated what the Pasteur Institute had isolated. And that's why he did not receive the Nobel Prize, along with Montagnier and Barré-Sinoussi. Truth or Dare says, I keep seeing no live virus found in milk. Is this lack of knowledge? Do they mean virions or replicating virus? Seems meaningless to me. So most of the detections are done by polymerase chain reaction, right, which just measures pieces of viral genetic material. And I believe a few people have done infectivity assays on the milk, and they don't find any infectivity. So I think there's very little, if any, infectious virus. Remember, there's always a limit of detection. You can't say there's zero infectivity because your assay is always limited, but they're not detecting any infectivity by the assays they're using. And that's good. I mean, the real concern is not the milk because it's pasteurized, but it should be pasteurized. But, people who work with cattle, they get close to them, especially to milk them, right? They get right up there to pop on the pumpers. And yeah, I've seen movies of that. So so they get close. So they can get infected and then transmit it to other people. That's the concern, right? It's not really the milk, unless you want to drink unpasteurized milk. But apparently there's no infectivity there.
Okay, I think you just got me in trouble, so now I need to defend myself.
Okay, go ahead.
All right. So Peter says scientists are not typically neurotypical. That's what makes us us. And I absolutely buy that. But I also have said on numerous occasions, artists are not neurotypical. I don't really think there is a neurotypical, and I wish some scientist would prove it. But ultimately, I see very little difference when I'm on the road with you and meeting the scientists and the baby scientists and between an artist personality and a scientist personality. It's just a direction that people end up in. And when I say Vincent needs to act more human, it's because oftentimes he is put on a pedestal by a lot of people out there that don't recognize that he is a regular person too. And so I encourage things like videoing him dancing because he's a good dancer and he likes to dance, but it's not something many people get to see very often. And it makes them understand that he's a regular person also. Even though he just tried to get me in trouble by acting like I don't think scientists are people.
No, I wasn't trying to get you in trouble. I know we've had this discussion before, and I just wanted to bring it up.
Mark said, "Oh my, Karen."
Yes. Let's see. Pete says unscreened blood products to hemophiliacs in the UK continues as an unresolved scandal. No government has bitten the bullet of compensation. Yeah, so in the beginning of the AIDS pandemic, a lot of unscreened blood, unscreened for HIV-1 was given to people, and a lot of people got AIDS from it, and they shouldn't have. They had zero risk factors except that was their risk factor. So the companies would not admit that it was a problem until they developed a blood test and they could finally test the blood. So that's a real scandal. Right? That's just horrible that they wouldn't believe it. So science is not about belief. It's looking at the data. And, it's like cigarette companies not looking at the data, hiding it and so forth.
That's because money is more important than human lives.
Yes, that's unfortunately true.
But also they just lifted the you can't donate blood if you've had sex with men before rule too, didn't they?
They can't donate blood if you lived in the UK during the BSE outbreak. That's for sure.
But in the U.S., you used to not be able to donate blood
that's right.
if you were a man who had sex with men.
That's correct.
And I found that personally offensive because everybody else went in and they didn't ask us if we had had sex with men who had had sex with men. But they based it again on sexuality instead of sexual activity, and they didn't ask enough questions about other people to find out if they were just as or more at risk. And even men who had sex with men, I had friends who hadn't had sex in 15 years, right? Mm-hmm. Still weren't allowed to donate blood.
Not allowed, yeah. So Garrett writes, H5N1 was devastating to marine mammals. That's actually a concern. We had a TWIV in Baltimore last year, and Ron Fouché and some of the other panelists said that was their main concern, not humans so much, but it's infecting lots of marine mammals, and they're worried about their populations. Yeah. Visto asks, could HIV have been in human, small, isolated populations for millennia? The clock analysis, looking at the evolution of the virus, right, says it's not earlier than, the late 1800s, so not millennia. SIV has been in monkeys for millennia, probably 30 to 40,000 years, but not HIV in humans. Oh, so John asks Visto, do we know how old SIV is? About, some estimates have it at least 30,000 years old. And so it's been circulating in old world monkeys for many, many years. Truth or Dare says, speculation on social media. What's that? What is social media?
Social media? It's that thing. It's you have a TikTok, Vincent.
I know. That's social media. We have Instagram, we have Facebook,
yeah.
we got
you even have
everything.
personal stuff.
Isn't YouTube social media?
YouTube.
Did
you just call it YouTube?
Isn't that social media?
It's a video platform.
Well,
I guess it could function as
but they're chats.
Media.
Like we're on YouTube now, and there's a chat. It's interactive.
Right.
Mark says, Karen, we need video of Vincent dancing. Well, you have it.
I do, but it's not good because I didn't have good lighting. So I'm waiting for ASV
Actually, it's
this
quite
year
good.
again.
You should release some of it.
Okay, I'll do it.
It's really good. I like it. All right. So, truth or dare? Speculation on social media that viral fragments in dairy could confer a measure of immune response. Comment. I don't think so, because to make immunity, you need to have some protein, some viral protein, and these are fragments of viral genomes, and the likelihood that they're going to be translated into protein is low. It's not zero, but it's low, and plus there's not much of it there, so I don't think it's going to confer any immunity that's of any value. Yeah. Now I have to see, Tony says, what the other lentiviruses are. There are lots. As I said, there are horse and cow and cat and goat and others. You can just look it up. Yeah. And they've been known for years. So this is nothing new, except that it was new to people. Garrett says H5 was devastating to marine mammals, but cows don't have the same levels of mortality. Any reason for this? Good question. We don't know, and it would be very hard to figure it out. It's not easy to do experiments with cows. They're big and expensive, but there could be some form of immunity in cows. The ability of the virus to reproduce in cells could be much lower in cows than in marine mammals. You could do some experiments with just cells to look at that to get a clue. But it is an interesting question, right? Because you'd like to know, can we learn something that could help us by
You could
seeing?
submerge the cows in water and see if that makes them more susceptible.
Why would you put them in water? You mean with their
'Cause
heads
it says
sticking out?
It was devastating to marine mammals.
Oh, you see, the water is the what you have to do in salt water, right?
It's a real good thing I'm not a scientist.
Good thing you're not a scientist, yes. But this is a non-scientist question. It seems very obvious that you could do that because the water jumps out as the big difference, right? But it's a hard experiment to do. You can't. I'm picturing you put a cow in a cage and lower it into the ocean so its head is above. No, we can't do
I
that.
feel like it would be too traumatizing for the cows.
It's too traumatizing, and you couldn't get approval to do that experiment, which is good.
PETA would have a meltdown.
G says there was a phrase back then: when you sleep with someone, you sleep with everyone they had sex with. Anybody remember? Add those of us with bleeding disorders were freaked out. I don't know that, but thank you for sharing. I don't remember that. Hello from Austin, Texas. Yes, Garrett. Nice to see you.
I'm sorry, G. I remember that.
Did we meet Garrett?
No. I remember the
The
phrase.
saying, okay.
Yeah.
Artemis from Montreal. Why can't the T cells be cleaned of the virus? Okay. So they could, and that's one of the approaches. Because as you know, the viral DNA gets integrated into the chromosome of the T cell. And these are memory T cells, which live many, many days. And then before they die, they divide, and then another one with the HIV DNA, and it lives for many, many days. So you see, you can't get rid of them. People are trying all kinds of things to get rid of the viral DNA. So one thing is, let's turn on all the T cells in someone so that they make virus, and then hit them with antivirals. So the virus will kill those T cells, and then you inactivate virus with an antiviral. So the problem is you have to be able to get all the T cells to start producing virus at once. If there are a few left, it doesn't work. That's called shock and block. Oh my gosh, the names they have for these things. Shock and block. Because you shock the T cells into making virus and then you block infectivity. I think a better approach is to cut out the viral genome from T cells or cut out the receptor that the virus needs from the T cell genome. Now, you may remember a couple of years ago, a scientist in China did this to embryos. So there was a couple in China who wanted to have kids, but I think one of the two was HIV positive. So they didn't want to have a child that would get AIDS, which, by the way, what you have to do is take retrovirals before birth, and that solves it. But they convinced the scientist to take out some of her eggs, cut out the receptor, one of the two receptors for HIV from the genome of the egg by CRISPR, and then putting it back into her, and then she gave birth to twins. And those twins lack the receptor. It's called CCR5. But apparently they have other issues. And the guy, of course, got in big trouble because he didn't get permission to do that. We're not allowed to edit people's DNA right now in ways like that. And there's only one genome editing procedure that's licensed, and that's to cure, I think it's hemophilia or hemoglobinopathy, where you take out stem cells and you cut out the defective gene, you replace it with a good one, then you put it back in people. That's the only thing that's been approved.
So they went digging around in her egg. She went digging around in her own egg and removed the capacity for them to actually even get infected?
Yes, they took out a receptor. So then all the cells...
And then made a weird mess out of other stuff?
Well, apparently they have other issues, but we don't get much news about it, right? So I don't know exactly
Yeah,
what's
you wouldn't
going on.
want to make that public, probably.
Well, they it was very publicized. Oh, yeah, the problems, yes.
Right,
yeah,
right.
They're not going to make it.
Wow.
So that's why it's hard to clean the T cells, but they would like to, because I think a vaccine is going to be highly unlikely. Why? Because it takes about 2 weeks from the day you're infected to establishment of this T cell latent reservoir. And then after that, that's it. You can't do anything about it. You take an antiretrovirus, you don't get sick, but the virus genome will still be there. So you have 2 weeks. No vaccine prevents infection. So any HIV vaccine you make, you're still going to get a little infection, and that's going to integrate. And so you need to make a sterilizing vaccine, which in my opinion is probably impossible. So the alternative is to somehow devise a very easy way to cut the gene out of either the virus DNA or the receptor out of people. And it's what people are working on. Abdul Rahman says, I highly appreciate how you keep reminding people about AIDS still being a pandemic. The cell-to-cell transmission. Can other zoonotic viruses develop that capability? Yes, there are certainly other viruses that can fuse cells. Coronaviruses can cause cell fusion, and other zoonotic viruses, paramyxoviruses. So, not paramyxo, sorry. But yes, and that is insidious, right? Because then the virus can go from cell to cell, escaping antibody. Jill says, my high school age kids don't even know what AIDS is.
Jeez.
It's interesting. I think everyone should be taught in school at some point. Probably, I don't know, high school? Do you think high school is a good time?
Well, I mean, we used to tour junior high and high schools. You have to talk about sex in order to talk about HIV. And you can't, there's no way to have a conversation in any space that's like abstinence space. So that takes out some of the places kids are learning. But beyond that, Indianapolis used to be years ago, every 8 years it would become the syphilis capital of the nation. So we would get money then from the government to go into transition classes in prisons and high schools and warn people.
Yep.
Tell them how to protect themselves.
Les says, clotting factor drug was made by combining many, many donations. That's correct. And they would pay people to donate blood. And often you get people who are kind of down and out, and they need the money, and their health isn't great, and many of them had AIDS. And so you had an overrepresentation of HIV in these blood donations. And yes, they were all combined, and so you had a real problem there. Really high incidence of AIDS in populations, particularly hemophiliacs that need frequent blood transfusions. They got it in very high numbers. Yeah. Okay. The mammals are probably consuming infected birds. Yeah, to get infected with H5N1. The birds that are infected with influenza H5N1 are excreting the virus in their poop, I guess. What do you call the bird excrement? Is there a word for that?
Bird poop.
Bird poop. Thank you. I have my expert here to tell me what bird poop, and the virus falls from the skies, basically. So if you've seen pictures of cars covered with bird poop in certain countries because they parked them under a tree, it's probably some H5N1 influenza virus there, right?
I just want to hear you say bird poop a few more
Bird
times.
poop. Bird poop. I wouldn't think that wild marine mammals are a great comparison model for domestic bovines. No, but they're all problematic, right? They're all sick, and we don't want them to be sick, not only for their own health, but they're going to transmit it to other animals, including humans. Yeah. Les says, blood banks did not test for Hep virus correlated with HIV. They had no test for HIV, but they were warned that their blood was a problem, and they said, no, it's not a problem, and they just kept on selling it.
Mm-hmm.
Hello from the Philippines. That's a distance away. Peter says, I've known a couple of troglodytes. Yeah, we all have. We all have. My dad uses this word for me and our pets. So now I don't know what word this is referring to, but there it is in the stream forever.
Smokey the Bear is his
yes,
icon.
that's right. Daniel Griffin said the first thing he said, no immunity would be conferred from inactive virus particles in milk. Yep. Is the origin to the Caribbean to New York City an example of genetic bottlenecking? To a certain extent, I guess, because it's, you're getting a low inoculum, right? It's not a huge, diverse population of viruses. So by definition, you take a subset of the virus in that infected person. So an infected person with HIV has something like 10 to the 10th, 10 to the 11th particles at any one time, virus particles. And then maybe 10,000 are getting into the next person. So that's a bottleneck. But then in that person, the virus expands, and so the bottleneck is relieved. So there's no problem. The only issue is the following, that let's say, there are many subtypes of HIVs, and if the person came from a country with, say, subtype B, then wherever that person initiated the infection in another country, subtype B would predominate. That's how it works. And by the way, there have been very notable criminal cases where people were accused of spreading HIV/AIDS intentionally. And they used genome sequence analysis to prove that certain individuals did, in fact, infect all of these people who were HIV positive. They got the genomes from all of them and from that person, and they could prove with phylogenetic trees that that person definitely gave them the virus. A key finding in court trials. Very interesting. Tony wonders what the drug regimen is for HIV-positive infants. I don't know. I'm sure a physician might know this, but I do not know. Martin, think of all of us all the time swimming in an ocean of microbes, including viruses. That's the truth, I fear. Yes, we are. But, Mark, we're mostly healthy, right? Someone asked Nell Zeldy today on Tweevo, should we trust viruses? And Nell says, of course not. Look what just happened. Why would you trust the virus? On the other hand, sometimes they can help, right? All these endogenous viruses that give us genes that are useful, like for our placenta. Is the mechanism for monkey resistance to HIV known? Yes, there are some mechanisms. I cannot tell you what they are, but yes, they are known. They involve overcoming restriction factors of the virus. So,
and using condoms.
Monkeys don't use condoms. That's the problem.
What?
That's why all the non-human primates are infected. But yes, and that's well known. And also the transfer into humans, what enabled that to happen has also been well studied, and I don't have them at my fingertips here, but that's a really good question. Markle says, Arthur Ashe, a famous tennis player, got HIV from a blood transfusion during heart surgery. Yes, that's absolutely right. That was in the day when We didn't know. We weren't testing. You
Yeah,
got AIDS.
I'm actually Ryan White generation, and I can tell by some of these comments, quite a few of these people are too.
People said they can't hear you, so I turned up the volume again.
Oh,
Did Isaac Asimov die of or get AIDS? Did he? Let's look it up.
mm,
I should have you do this.
I am doing it, but
Yeah.
you can't stop yourself.
Asimov had triple bypass in 1983 at NYU. He got HIV from a blood transfusion. He died in 92. I don't know if he died of AIDS or something, because maybe there was triple therapy, not then. I don't know. Yes, a lot of people got it from blood transfusions. Let's see. John says the 12% divergence in the envelope gene could have occurred in chimps, not in humans. This redates the origin in humans to the 50s. The chimps were brought to the Congo to support polio research. Okay. I know that the chimps were I don't know if they were brought there. They were maybe from elsewhere in Africa, but they were certainly used for polio research in Africa. And for a while, remember the Origin of AIDS book? Forgot who the author was, but he said that HIV originated from the polio vaccine trials in the Congo, and that's not true for reasons I don't have time to go into, but I was involved in disproving that actually many years ago. Jessica says, depends how deep the cut is. Accidental needle sticks are a concern, right? So needle sticks, the cut hunter wouldn't have a needle, but he or she could have a deep cut, and then the blood could flow into it from the chimp. Yeah.
Yeah, see, back in the 90s, what they were telling us is that the needle sticks were more dangerous because the pipette or whatever on the needle keeps the blood in it from
Yes.
being exposed to oxygen. And I have now Googled it, and I'm not crazy. That actually was theories for a while. So
Yeah, but it doesn't last seconds. That's the thing.
they did used to tell us that.
Yeah,
though.
I don't doubt it. Patricia says that's why needle sticks are something to take very, very seriously, especially with the larger bore needles instruments. Yeah. So if you're working in a lab and using HIV or an HIV vector and you stick yourself, you should immediately get antiretroviral therapy, because you will prevent establishment of a latent reservoir, because you know the moment of your needle stick, right? The potential HIV inoculation. And so often, if you have sex or use intravenous drugs, you don't know the one time when you got infected until you have symptoms, and then it's too late. But that's why PrEP is good, because if you are going to do intravenous drugs and if you're going to have risky sex, you should take PrEP, and then you will have a good chance of preventing getting infected. Artemis says, why couldn't it be eating bushmeat with animal blood in it rather than a cut hunter? Because the bush, so that's certainly the case for Ebola virus, right? They think people eating the bushmeat, but you don't acquire HIV by eating infected material, right? It's inactivated in your stomach. I would never say never, but it's less likelihood.
Vampires aren't real susceptible unless they have cuts in their mouths too.
That's right, cuts in the mouth.
Right.
So, and also the hunters would bring the carcass to market or home where it would be sold or cooked, right? So they don't kill the chimp and eat it right there. They butcher it, and the butchering is a risky process because they have big knives, right? So that's the idea.
It's probably not that risky for good butchers.
Yeah, well, I don't know if these hunters were good butchers.
Yeah.
But everyone cuts themselves, right? Even butchers in meat shops cut themselves. So,
Maybe.
Blood is routinely considered a biohazard due to the possibility of viruses. Yes, it is a biohazard, especially if it hasn't yet been tested, right? Yes. How similar are HIV and FIV? I would guess there are not. I mean, you could see that they are both lentiviruses, but there would be over 20% divergence in the protein sequence. So, but they have envelope, they have GAG, They have polymerase and they have accessory proteins as well. I heard something called vampire facials, and some women got HIV recently from a place offering the service, which I imagine any health codes would have shut it down. Well, you put blood on your face, I guess? Yeah, you could. That would be a problem, depending on the source of the blood, right?
Wouldn't you need to have some way for it to enter your bloodstream?
Cuts. Yeah.
Maybe they exfoliated strongly beforehand.
But, yeah, I suppose that could do it. I don't know of...
or it could get in your eye, I guess, right?
Yeah, I suppose it could get in your eye. I'm not aware of this, but I would be interested in any case reports on that. Okay, Denise, I already listened to Vincent's entire lecture on this for his course at Columbia this past week. It informed me to be able to advise my physician associate family member who her turkey hunts in Wisconsin. That was about avian influenza virus. Okay. Yes, thousands of partners. Yes. Well, so he said, right? There's a bit of bravado.
Who?
Gaetan Dugas. Do you know that name?
No.
Go Google it. You'll learn some things. Well, you always learn things at the incubator, but he was a flight attendant, flew all over the place, and had a lot of sex. He claimed he had thousands of partners. Yeah. Yep. Denise said, "Yeah, I don't think I'll tell anyone I was once an international flight attendant again. Don't worry. I don't think it's a problem for most people."
Who's Canadian?
Okay. There are flat earthers on the internet saying the earth is flat. So, absolutely. Absolutely. Denise is concerned for the people of my home, the dairy state of Wisconsin, where dairy union is tight-lipped and currently turkey hunting is also happening in fields of farmers. Ugh. I don't know how concerned you should be, but yes, as I said earlier, I think we can handle an outbreak of H5N1. So I don't think SARS-CoV-2. The Crimson Frenzy asks, what are the strongest virulence factors in this virus that is gene and gene product? I don't know which virus. I guess we've mostly talked about HIV-1 tonight. So there are some accessory proteins that block host cell defenses, and those are the highest. Those are the virulence factors. If you take those out, then the virus loses virulence. But remember, people don't die of HIV infection. They die of opportunistic infections that occur because your immune system is severely suppressed and you can't deal with Candida or CMV or pneumocystis.
I'm sorry, I wasn't laughing at that. I just keep accidentally reading Visto Tuti's comments,
Yes.
and I'm having a hard time controlling myself.
200 people tonight. Wow. All to see Vinny. All to see little Vinny. How about that? Hit the like button, folks. 157 likes. We would love to have other people come.
Have they seen Little Vinny?
Have they seen it?
Yeah.
Well, it's just a telephone, right? Now, we have a telephone here at the incubator. We have an official phone, which is an iPhone. And it's called Little Vinny. It was not my idea.
Whose idea was it?
It was your idea, Karen.
No, it wasn't.
That's fine. Well, you didn't ask me. You just said, this is Little Vinny. Okay. How could you reject someone naming a phone after you?
It's the point in which I needed to download your brain into something else.
Yeah. Anyway, 120 likes, 193 viewing. All right, now the likes are going up. Thank you. Abdul, thank you for your support of science communication. Really appreciate it. Robert says chimps hunt. Where did Robert go? Oh, my gosh. Here we go again. Robert, chimps hunt and eat monkeys, so I understand how SIV got into them. How did it get into gorillas? I thought they were herbivores. They probably fight chimpanzees, right, in blood exchange. That's the theory. So, but who knows, right? Elise says, we're at this stage now where everything jumps around when I click on it. I was an OR nurse back then, and we would wear eye protection in case of blood splatter into our eyes. Prior to AIDS, dentists did not even wear gloves, but started to when AIDS came. Yes, for sure. Okay, Mark Martin says the CCR5 deletion that impairs HIV binding, the deletion allele is more common in Northern Europe, about 12% of descendants of Northern Europeans. Does that mean something like HIV swept through Europe a couple of thousand years ago? So something swept through that applied the pressure,
The
right?
Black Plague?
So it was thought to be the plague, and people looked at that, and it is not related to plague. People thought it might be smallpox. It's not related to smallpox. So we don't have any idea what the selection was at the moment, Mark. It's a very good question. Very good question. Now we're getting all these cool questions towards the end here. Did I cover the mRNA trial for an HIV vaccine? No, I didn't talk about it at all. And it's one ongoing. I don't even know. But I know that the results haven't been released yet. So Denise writes, no, I'm sorry. I clicked on Denise and then Patricia popped up under
So
the
if
thing.
HIV was transferred from an animal vector to hunter, why couldn't it be more of an issue in America's white-tailed deer population to those hunters, as Dixon suggested?
Well, deer don't have HIV. Are you thinking of? You're thinking of chronic wasting disease of deer, and why aren't humans getting it? And they're not, because the prion from deer doesn't seem to infect and reproduce in people. So that, for now anyway, it could change, but no hunter seems to have contracted a prion disease from deer so far. Okay, now Physate Liu has a bunch of questions, which I hope I can answer. I will be a graduate of biotech in the fall. I have some questions of mRNA vaccine LNP for you. All right, first, the pharmacokinetics of lipid nanoparticles, which specific cell type will intake. So the antigen-presenting cells primarily take up the lipid nanoparticles. How does the antigen get presented? So it's translated in the cytoplasm into protein. The protein is processed by the proteasome. It's displayed on MHC molecules on the surface, and then they go into the lymph node and present it to T cells in the lymph node to get the immune response. Let's see where the next question is here.
What is the outcome of cell that intake and express exogenous mRNA? Will it be killed by... Did you already do that one?
No, I didn't, but where is it?
It's right under the...
See,
Oh,
when
it's
I
1
click
and 3.
now,
No,
towards
no.
the end of a stream, this is what happens. I lose.
see 1 and 3. I see 1, 2...
You know, it would be helpful when I start answering a question, Karen. Can you? Oh, you don't have the stream there. You can't.
No, but I can read it for you. Or you could, I don't know. We have to figure out, there has to be technology for me to be able to pull things up for you. And it not
here
just
we
be
go. I
one
found
person.
it. I'm not even going to click on it because that's the problem. If I click on the star, what is the outcome and income of here it is. Now when I star it, I can find it quickly. What's the outcome of cell that intakes and expresses? Will it be killed by T cells? No, antigen-presenting cells are not killed by T cells. Macrophages and dendritic cells present antigens, and that's what they do for a living. And the T cells do not kill them. The T cells get activated and proliferate, and then they go out into the periphery to kill infected cells, but certainly not the antigen-presenting cell. What would the saying be? Don't kill the hand that presents you, something like that?
Bite the hand that feeds you?
Don't bite the hands that feed you, for sure.
You know what I can do is I can pin them to the top, but I don't know if I do it
I might not see it here.
Yeah.
Because, it's funny that in the later parts of the stream, when I click on a comment, like I just clicked on Physate Lose to show it up on the window, then it pops all the way up in my stream, and I have to find it again. Let's see. I don't know the answer. What is the best pizza in New York after di Fara? I have no clue. Do you have any clue, Karen? You don't eat pizza.
I eat pizza all the time, and I just think this whole idea that I've had more people come into town and be like, "We need a New York pizza," and as far as I can tell, New York pizza is like cheap by the slice, thin that you can eat walking down the street. It's not. I mean, I don't know the big hoopla about any of it.
John, thank you for your contribution to science communication. So John popped up, but then I could find him because it's colored green on my chat here. We appreciate. Microbe TV taught me that nature is the world's biggest lab. It's true.
Okay, you see, somebody's asking about Angela's podcast and when it's going to make its debut. Tona,
I don't know when it is. Do you?
I don't think we have a deadline yet. We're working out some kinks, I think, and they're getting their groove going, and we'll let you know.
I'll let you know. There's some funny comments here. They keep popping around. Holy cow, why are they doing this? Maybe if I make the window larger.
Ooh. I can't pronounce names, but is Aerozone?
Aerozone.
Yes, Aerozone says apparently cicadas urinate in a direct downward stream.
How would you know that?
It doesn't matter. It's cool, Vincent.
CRISPR to cure sickle cell. I don't remember what the blood. I just taught it in my lecture. So let's bring it up. Let's open my lecture from there. That would be therapeutic viruses, and that was the last lecture of the course. And I gave a list of some approved gene therapy modalities. That's lecture 25. Here, let's bring it up. And it's one CRISPR that has just been FDA approved. Let's find it here. Here we go. Sickle cell disease. Thank you. Yes, sickle cell disease. Thank you, Tom. You knew the answer. Yep. Leo says, I'm going in for knee replacement next week. Part of the pre-op test was for Staph aureus, which came back positive. Yeah, they don't want you to have that in your joint. I asked what the CT value and was told that it's only for SARS-CoV-2. Well, I don't know that they do it by PCR. Maybe they did a culture. I'm not sure. It's, out of my wheelhouse, microbiology. Yeah, the sickle cell is a CRISPR modification. It's approved. Yeah. You take cells out of the patient, you modify them, and then put them back. What kind of contact are people having with cow excretions? I don't know. If you're next to a cow and you can get contact with all kinds of excretions. Maybe you pet them. Maybe you say hello and you talk to them. I don't know. They breathe on you, right? I'm not sure. You have to ask a farmer. Yeah, so the office hours with Paul Benash had a lot of research on HIV. If you missed that, that's a recent one. Siddhartha from New York, welcome. Hello again. Guano. Yeah, bird poop is guano. Karen, you didn't know that, huh?
I thought bat poop was guano.
Yeah, seabirds and bats both.
Birds and bats have the same kind of poop?
Yeah, bird poop is called guano. It used to be collected and used as fertilizer, yeah.
You think that's from a time when people thought bats were birds? Or are bats birds?
No, they're mammals.
That's what I thought.
H5 seems to be more deadly to seabirds than to inland birds. I'm not sure about that. I think it's very hard to compare virulence in different species, and you're depending on observations. So I would defer. We're going to have an H5 expert on TWIV at some point, so we'll wait to ask them about that. It's estimated that 20% of the HIV-infected people in Thailand currently don't know they are infected. It's really a big problem, right? That's how you spread it. Paul Farmer writes, HIV most likely originated in Africa, came to the U.S. and Europe, and was introduced into Haiti. No, did not happen that way. The phylogenomics clearly shows that it went from Africa to Haiti. Okay? That book is wrong.
I totally lost where you are on here.
I'm near the bottom. Should this year be a concern going swimming with those geese that hang around the lakes after TWIP? Pools are not even safe. Well, the key is to have proper chlorination, right? If a pool is properly chlorinated, you will not get infections. So, like, you never get Giardia from a pool because it's destroyed by chlorine at levels that do not destroy a cryptococcus, for example. But you don't know. You don't know if the pool is properly sanitized. So if you see geese in a pool before you go in it, forget it. Yeah, we talked about Dasak, before. I wouldn't say he got waxed. I think he waxed Congress in reverse. So WebMD says hours for HIV inactivation. That's closer to home. That's fine.
Okay, I'm going to have to ditch you because I'm going to go talk to some Australians.
Okay, goodbye.
But I need you to read out loud anything else that references poop or all of the comments of Visto Tutti. All right, please. Okay. While I'm gone, I'm just going to trust you to
Karen.
do that.
Goodbye.
Thank you all so much. Have a fabulous evening.
Exfoliating gives you micro cuts. There you go. What antibiotics can stop virus infection? Well, antibiotics are for bacteria. Now, a paper just came out in PNAS suggesting that Neosporin is a broad antiviral because it induces interferon-stimulated genes, which are antiviral themselves. So we'll see where that goes. But antibiotics do not have a direct effect on viruses as they do on bacteria. A lot of talk about facials and needling and all this stuff. Yes, if oral personnel get stuck while working on an HIV positive, they also check compatibility. They sequence the genome to see what kind of retroviral they're going to get. Because if a person had some resistance, you're not going to give them those antivirals. Yeah. What do blood vessels and epithelial cells of the human and monkey reproductive system have in common for HIV virus? CD4 positive T cells. Those are the cells that are the main target of HIV reproduction, and both humans and non-human primates have those cells. And why is it different from coronaviruses? Coronaviruses bind different receptors, H2 for SARS-CoV-2, for example, and those are not widely conserved among different animals, which is why the virus doesn't infect everything, although it does infect a lot. Yeah. If people with HIV get opportunistic infections, what are those infections called in HIV negatives? Well, if someone is getting those infections, they're usually immunocompromised, right? So, yeah, we would call them an opportunistic infection and an immunocompromised person because it's opportunistic in the sense that if you had a normal immune system, you wouldn't be getting these infections, right? Good questions. It always gets lively here towards the end. I learned here that mosquitoes aren't able to transmit HIV. It doesn't replicate in the mosquito. Yeah, most viruses that are mosquito transmitted have to reproduce in the vector. Yeah. Don't kill the messenger. That was what I was looking for. Yes. And Patricia confirms that opportunistic is used anytime the host is immune compromised. And Peter, I heart New York pizza. Jill likes your voice, Karen. A lot of discussion of opportunistic. That's interesting. And the topic of urination, if one gets buried in an avalanche, one is supposed to pee in the trapped snow to know which way is down and which way is up to make their way out. Well, that's very interesting. I'm not going to get caught in an avalanche. I'm never going to be in a position to do that, but it's good to know. That is just great. I love it. You know, did I read the article about CRISPR to treat senescence? So I know there's an association between endogenous retroviruses and senescence. So maybe this is doing something with those, removing those in some way. Siddhartha, thank you for your support of science communication. We appreciate it very much. I know the birds are aerial bombers on my car roof. Yeah. Asorius is systemic in about 30% of healthy people. That's right. And if you get a joint replacement, they really don't want it to be there because it may cause an infection, and then that's really bad. Then it makes a biofilm on the replacement, and it's really hard to treat. Yeah. New rule suggestions. Preface all questions with a poop emoji. Now I'm going to add the term poop to all my questions from now on. Oh, but people got me saying poop a lot. Gabriel, thank you so much for your contribution from Mexico. MX. See, that's the MX dollar. Thank you very much. You know, it looks like I have exhausted your questions. I did skip a few. Don't be insulted. It's just what I feel like I can answer in some useful way. But I believe all of you have had a lovely conversation among yourselves. Could monkey antiviral proteins for SIV be used at all? Viral gene therapy for HIV? That's a good question. I don't know. Yeah. If you had a clear-cut example and it weren't immunogenic in humans, that's the problem if you're going to give people proteins. But hopefully, one could make a small molecule that does the same. This is very interesting. Artemis says, for peeing when caught in an avalanche, it requires to be able to take some clothes off. Not men, you know. We can just flip a zip. But yeah, I understand that women can't just do that. Right. You got to drop your pants, and then you're freezing. You got that snow all over the place. Yep, Mexican dollar is a peso. Thank you for that. You know, folks, this is fun, isn't it? Let me thank all of our moderators tonight. We had Les here. We had Tom. We had Vanity Nutrition. We had, I saw Steph for a while, though she was very quiet. She didn't say much tonight. We had Andrew. Thank you all for coming. Did I say Vanity? Yes, Andrew, Les. Thank you for moderating tonight. I know that you are not without work to do here, and it's greatly appreciated that you keep this a civilized place. And thank you all for coming. So here, John says, Vinny Mod's listeners, he said this thing in Russian, which is I love you, I think, right? Thank you all for coming. We had over 200 people at some point. I'm very impressed that 200 people would come to hear me. So thank you for that. Heather says, had a coworker with a close monkeypox exposure. She was told she'd get vaccinated after exposure. You use some time before, before the rash, you can get vaccinated. Yeah. Yeah. And also for rabies, right? Same thing. Thanks, everybody. Let's see. Oh, thank you, Steph. Thank you for being here as well. Vanity. Okay, so now we have to say goodbye. Das vidanya, right? And also, poka, das vidanya. Arrivederci, ciao, bonsoir. And what is it in Spanish that you say goodbye? Spasibo tovarish. Claire. Spasibo tovarish. So spasiba is thank you. I don't know what tovarish is. Cool. I rock, really. Thank you, Tammy. Thank you very much. Oh, dear. Thanks, everyone, for coming. I don't want to let you go. So, poka. No, that's good night. Dobry večer. Dobry večer is like thank you or something like that. Spasiba. Hasta la vista. Hasta luego. Thanks, everybody. Au revoir. That's right. And folks, give your own goodbye here. I'd love to see them scroll by. Zaijan. Cool. Zaijan. Buenas noches. Shalom. This is a new one now. Okay, everybody say goodbye in your language. Dovidenya. Cool. Oh. Shalom. Hasta la vista. Hasta luego. Adios. I'll just sit here for a few minutes. What is the Sunday TWIV theme? I know one paper is going to be about parechoviruses, just to introduce people to parechoviruses and their receptor. Shalamehala. Shalamehala means goodbye. Oh, that's so cool. Auf Wiedersehen. Arrivederci is Italian, of course. TTFN, ta-ta for now. Om Shalom. La Hitraot. That I don't know. Choose informal. Finnish goodbye. Oh boy, that's fun. Any others? Let's give it a few more minutes. I want to see. Allah Hemane. Allah Hamana? Is that it? When we meet, you'll have to teach me how to pronounce Ma Asalama. Tata. Do Widesnia. No, do Widzania. There you go. Adio. Salamat. Don't take any wooden nickels. Yeah, TTFN is good. I always like that one. Good not. Goda not. So this is an amazing international audience that we have here. Moin, North German. Those Do Widzania is Polish. Got it. Yes, that sounds Polish. Do Widzania. See, I could sort of figure it out if I can read the characters, but I can't always. Guten buona notte. Guten Nacht. Lila Tov. This will be a permanent part of this stream. It was Yavas Lubliu. I love you. I wasn't going to use it myself. And Patricia says toodles. And Artemis says tot zin, until soon in Dutch. Tot zin. I don't know if you pronounce the S. Tot zins or tot zin. Karen is now on a Zoom call to Australia because we're trying to plot some travels there. Don has got this two characters that I don't know. Garrett, abiento. Toodles. It's very funny. Unless, don't know what it is. What's that, Les? What language and what is it? Tona said that was good. Oh, Liatov in Australian. Where did she say that? Yes, you pronounced the S in zins. Okay. This is a cool way to end a stream. Yeah, I think so. Good morning in Australia. That was Lila Tov, right? And then Vanity, Uzred Zaushanost. Latvian goodbye phonetically. Uzred Zaushanost. I love these Slavic languages, Cyrillic, and Well, I'm not saying the right words, but you know what I mean. It's cool. Bye-bye in Mandarin. It's great. Sounds phonetic, right? Bye-bye. Love it. Languages are the coolest, right? We are a Tower of Babel. It's Hindi for goodbye. So that interesting set of characters. That's Hindi. Wow, fascinating. Look how straight those lines are. Very cool. Good Nacht, Rima. All right, folks. This has been fun. We'll be back next week. Oh, here's Maori. Hera. Fascinating. Wow, there's so many cool languages. I love them. Let's see what next week's date is. It's Wednesday, the 8th of May. We'll be back with another office hours. I'm trying to get a guest on, Theodora Hatziano, and one of these days she'll come and talk to you. John says this, Anyongi Gaseyo. Cool. Give my regards to Broadway. It's not too far from here. Good night, good nacht, bonne nuit, ma asalama, salamat, ala hemane, and dobra noche. Bye. Ate luego, Portuguese. All right, folks, that'll do it for another Office Hours. Thank you all for coming. Till next week, be safe. Love you all. I can't say it in Russian. Poka. Bye-bye.